Human antibodies play a critical role in the fight against malaria, a disease caused by the Plasmodium parasite. Research into these antibodies has uncovered their remarkable ability to offer broad inhibition against the variable proteins expressed by the malaria parasite. This phenomenon highlights their potential as tools for therapeutic development and disease prevention. Here's an in-depth exploration of this topic, segmented into 10 comprehensive sections. Malaria remains a leading global health challenge, with hundreds of thousands of deaths each year. A key factor in its persistence is the Plasmodium parasite's ability to evade the human immune system. The parasite achieves this through its variable surface proteins, particularly the 1PFEMP1 family, expressed on infected red blood cells. These proteins constantly change their structure to avoid immune detection, creating a major hurdle for vaccine development. However, recent findings suggest that human antibodies are capable of broad inhibition of these proteins, a discovery that could reshape malaria control strategies. The immune response to malaria infection is complex. When a person is infected, their immune system produces antibodies targeting the parasite. Initially, these antibodies are specific to the current strain of the parasite, which limits their effectiveness against future infections. However, repeated exposure to malaria in endemic regions leads to the development of broadly reactive antibodies that can neutralize diverse strains of the parasite. These antibodies target conserved regions within the variable proteins, areas that do not change even as the parasite evolves to escape immune detection. One of the most promising aspects of these broadly neutralizing antibodies is their specificity for conserved epitopes on PFEMP1. These conserved epitopes are essential for the parasite's ability to adhere to host cells, a critical step in its life cycle. By targeting these regions, the antibodies prevent the parasite from attaching to blood vessel walls, reducing the severity of symptoms and preventing complications such as cerebral malaria and organ damage. This action not only inhibits the parasite but also reduces inflammation and associated tissue damage caused by the immune response. Another significant breakthrough is the identification of monoclonal antibodies derived from individuals with high natural immunity to malaria. These antibodies demonstrate remarkable breadth, neutralizing a wide array of PFEMP1 variants. Structural studies have revealed that these antibodies bind to conserved regions with high affinity, effectively blocking the parasite's adhesion mechanisms. This discovery has opened new avenues for vaccine design, where mimicking these antibodies' specificity and breadth could lead to more effective and long-lasting malaria vaccines. Despite their potential, challenges remain in translating these findings into practical interventions. The complexity of producing monoclonal antibodies at scale, their cost, and the logistics of deploying them in malaria-endemic regions must be addressed. Innovative approaches, such as leveraging mRNA technology to induce the production of these antibodies in the human body, are being explored. Such strategies could make these life-saving therapies more accessible and affordable. Furthermore, the interplay between antibodies and other components of the immune system is critical for their effectiveness. For example, the complement system works in tandem with antibodies to destroy infected cells and parasites. Understanding how these systems collaborate enhances our knowledge of malaria immunity and helps optimize antibody-based therapies. This synergy also provides insights into why some individuals develop strong immunity while others remain vulnerable, even in endemic areas. The evolution of Plasmodium parasites poses a continuous challenge to human immunity. Yet, the persistence of conserved epitopes within variable proteins underscores a vulnerability in the parasite's defenses. Broadly neutralizing antibodies exploit this vulnerability, revealing a potential Achilles heel in the parasite. Their ability to recognize these conserved regions suggests that Plasmodium cannot completely alter these areas without compromising its survival, a limitation that can be leveraged in therapeutic design. Field studies in malaria endemic regions provide valuable data on the prevalence and effectiveness of these broadly neutralizing antibodies. By analyzing the antibody profiles of individuals with varying degrees of exposure and immunity, researchers have identified patterns that predict protective responses. These studies are crucial for refining antibody-based therapies and ensuring they are effective against the diverse strains of Plasmodium circulating globally. The implications of this research extend beyond therapeutic applications. Broadly neutralizing antibodies offer a blueprint for understanding immunity against other rapidly evolving pathogens, such as HIV and influenza. Their study highlights the potential for cross-disciplinary approaches in combating infectious diseases, where lessons learned from one pathogen can inform strategies against others. The development of malaria therapies based on these antibodies could inspire similar innovations in other fields. The discovery of broadly neutralizing human antibodies against malaria represents a turning point in the fight against this devastating disease. These antibodies have shown the ability to overcome one of malaria's most formidable defenses, the variability of its surface proteins. By targeting conserved regions essential to the parasite's survival, these antibodies provide a pathway to more effective treatments and vaccines. Continued research and innovation are needed to translate these findings into practical solutions, offering hope for millions affected by malaria worldwide. The discovery of broadly neutralizing human antibodies against malaria parasite proteins has ignited a new wave of research and innovation in the fight against this deadly disease. These antibodies have revealed not just the vulnerabilities of the Plasmodium parasite but also the incredible adaptability and sophistication of the human immune system. The Plasmodium parasite's survival strategy hinges on immune evasion, enabled by the vast diversity of its surface antigens, particularly PFEMP1. Broadly neutralizing antibodies, however, target conserved elements within these proteins. These conserved regions are vital for the parasite's functions, such as adhesion to endothelial cells and immune modulation. By focusing on these weak points, the antibodies bypass the parasite's antigenic variation, providing a potent and generalized defense mechanism. The structural basis of antibody recognition has been elucidated through advanced techniques like cryo-electron microscopy and X-ray crystallography. These studies reveal how antibodies latch onto conserved regions with remarkable precision. Understanding this interaction at the molecular level is critical for designing therapeutic interventions. By replicating these structural interactions, researchers aim to engineer synthetic antibodies or immunogens capable of eliciting similar broad-spectrum immunity. Human immunity to malaria often takes years of repeated exposure to develop, especially in endemic regions. Broadly neutralizing antibodies are typically found in individuals who have survived numerous infections. These antibodies are often the result of affinity maturation, a process where the immune system refines its response to better recognize conserved epitopes. Studying these naturally immune individuals provides a roadmap for developing vaccines or therapies that can confer immunity without the need for repeated infections. One promising approach is the use of antibody cocktails. Since malaria parasites exhibit a wide range of antigenic variations, combining multiple antibodies targeting different conserved epitopes can enhance therapeutic efficacy. These cocktails provide a multi-pronged attack, reducing the likelihood of the parasite developing resistance and increasing the durability of protection. This strategy has already shown promise in preclinical trials. The challenge of malaria eradication lies not only in treating individual infections but also in halting transmission. Broadly neutralizing antibodies can play a crucial role in this regard. By preventing the parasite from binding to host cells, these antibodies reduce the parasite load in the bloodstream, minimizing the likelihood of transmission to mosquitoes. This dual action, therapeutic and transmission blocking, makes them a vital tool in eradication campaigns. Antibodies targeting malaria proteins can also be integrated into passive immunization strategies. In this approach, high-risk populations, such as pregnant women and children, receive periodic infusions of these antibodies. Passive immunization offers immediate protection, bridging the gap until vaccines or other long-term solutions become widely available. Advances in antibody stabilization and delivery methods are making this approach increasingly feasible. Emerging technologies, such as mRNA-based platforms, offer exciting opportunities for deploying broadly neutralizing antibodies. By encoding the genetic instructions for these antibodies into mRNA, the body can produce them directly, mimicking natural immune responses. This approach, already successful in COVID-19 vaccines, holds immense potential for malaria. It combines the precision of antibody therapy with the scalability and adaptability of mRNA technology. The role of broadly neutralizing antibodies in severe malaria cases is particularly noteworthy. Severe malaria, characterized by complications like cerebral malaria and severe anemia, results from the parasite's aggressive adherence to endothelial cells and erythrocytes. Broadly neutralizing antibodies disrupt these interactions, alleviating symptoms and preventing disease progression. Their therapeutic application in severe cases could significantly reduce malaria mortality rates. The evolutionary arms race between humans and malaria parasites has shaped the immune system's ability to produce these powerful antibodies. Understanding this co-evolution provides insights into why certain populations exhibit higher resistance to malaria. Genetic studies have linked specific immune system genes, such as those encoding for antibody diversity, to enhanced immunity. These findings could guide personalized medicine approaches, tailoring treatments to genetic profiles. Finally, the potential for combining broadly neutralizing antibodies with other malaria control measures, such as insecticide-treated bed nets and anti-malarial drugs, represents a holistic strategy. Antibodies provide a biological layer of protection, complementing physical and chemical interventions. This integrated approach addresses malaria's multifaceted nature, tackling the disease from multiple angles and increasing the likelihood of achieving global eradication. In conclusion, the study and application of broadly neutralizing human antibodies represent a transformative leap in malaria research. These antibodies' ability to neutralize diverse strains, disrupt key parasite processes, and block transmission underscores their significance. With continued innovation and collaboration, these discoveries bring us closer to a future where malaria is no longer a global health threat.